Howdy my future doctors, welcome back to yet another video. Today we are going to discuss about the first week of embryonic development. Okay, gestation, the total duration of gestation can be divided into two periods, embryonic and fetal. And as you can understand from the name, the embryonic period is when the embryo develops and the fetal period is when the growth of the fetus happens. And this embryonic period is again can be classified into two periods, germinal and embryonic proper. Here also you can understand that this germinal means development of germ layers, and this embryonic proper is the proper development of embryo, the time in which the proper development of embryo happens. Now talking about the time periods, the embryonic period is from 1st to 8th week and the fetal period is from 9th to 36th week or 38th week. Okay, germinal, within this 1 to 8 it's 1st to 3rd week, and the embryonic proper is from 4th to 8th week. Okay, now as I told you earlier, we are going to discuss about the first week in this video. So the two major events that happening in this first week is cleavage and implantation. Okay, wait, you have heard about some morula formation, blastocyst formation, that sort of things. So where are they? Actually, cleavage has three subdivisions: compaction, morula formation, and blastocyst formation. So these are the three subdivisions of cleavage, and also there is implantation. Now let us look at cleavage. Cleavage starts right after 24 hours of fertilization and it lasts for 6 days. So after fertilization it will last till the 7th day. Okay, and what is cleavage? Okay, as you can see, this is the zygote, and the zygote is pretty big, right? And the zygote has a huge cytoplasm to nucleus ratio. Now we'll have to make sure this cytoplasm to nucleus ratio reduces. How? Through cell division, and reaches a cytoplasm to nucleus ratio that a normal somatic cell has. So that's what's going to happen. The cleavage is the process of repeated mitotic division of zygote within the zona pellucida. This is the zona pellucida. You know what is zona pellucida? Zona pellucida was the covering of the ovum, but now it is the covering of the zygote. So the process of repeated mitotic division of the zygote within the zona pellucida in rapid succession, giving rise to increasing number of smaller cells, which are blastomeres. Okay, and as you can see, after the first division, there is the formation of two daughter cells, but here one is bigger and the other one is smaller. Okay, so when the second division happens, the bigger cell will divide, and following that the smaller cell will divide, giving us a 4-cell stage. Okay, this 4-cell stage will again undergo division or cleavage, making an 8-cell stage. Now from this 8-cell stage, compaction begins. What is compaction? As the name suggests, the cells become compact and will be closely arranged to each other. Along with this compaction, when the division also or cleavage also takes place, we'll get a 16-cell stage called morula. In this stage, there will be the persistent zona pellucida, and there will be the cluster of cells within the zona pellucida which appears as a mulberry, or which has a resemblance to the appearance of a mulberry. Okay, if you take a cross section of this particular cell or this particular group of cell, the morula, we will see something like this. We can see there is an outer cell group which is called the outer cell mass. So straightforward, outer cell mass. And what is the inner cell group called? You guessed it, inner cell mass. Okay, Now what happens is this, any cell, if it want to undergo multiplication and growth, it need to have some nutrition, right? So initially this nutrition is the meager store of nutrients which is within this blastomeres, but as this multiplication progresses, it is not enough. So what happens is the endometrial secretions or the uterine milk will penetrate zona pellucida and will come into this morula. Now as the uterine milk comes in, it forms confluences with the interstitial spaces, and it will also make the inner cell mass more compact and will push this outer cell mass into the periphery, furthermore making it a bit flattened. Okay, so consequently what happens is there will be action of trypsin-like enzymes which will act on the zona pellucida. This trypsin-like enzyme is released combinedly by the blastomeres and also the endometrial lining. So this will affect the zona pellucida, and it will degrade the zona pellucida during the 4th to 5th day after fertilization. Okay, now what we have, the structure now what we have will undergo cleavage and will attain a 32 to 64 cell stage, which is known as the blastocyst. Now as you can see here, the morula is turning into the blastocyst. It is not like we can draw a margin and differentiate between certain structures very clearly. The margin between them is very blurred. Okay, so we cannot say, okay, this is definitely one turn of a switch and the morula is boom, blastocyst. No, it's not like that. There will be transitions. Okay, so as you can see here, the uterine milk will be filling this cavity, and the cells will be pushed to the periphery, and the outer cells will be pushed to the periphery, and the inner cell mass will be collected to a position which is called the embryonic pod. Okay, the embryonic pod. Now this outer cell mass is called the trophoblast. Okay, tropho means something that related to nutrition, and why we are calling this as trophoblast, because it will form the chorion, or basically the placenta of the future fetus. So it will help in the nourishment of the fetus, so that's why we are calling it as this trophoblast. And now near to the embryonic pod or the animal pod, we can see a group of trophoblasts which are called polar trophoblast, and opposite to that region, opposite to the embryonic pod, is the abembryonic pod. Also, all the other cells, all the other outermost cells, the trophoblast, are called mural trophoblast. Okay, now this outer cell, the trophoblast, will again be flattened, and also the inner cell mass will now be called as the embryoblast. Okay, this is the trophoblast. Now this with a blastocele is called a blastocyst. Okay, so the characteristic feature of the blastocyst is flattened trophoblast, an embryoblast on the embryonic pore, and a wide blastocyst. Okay, and as we said, this is the embryonic pole, and this is abembryonic pole, and this one is the polar trophoblast. This trophoblast are mural trophoblast, because it's the trophoblast cells other than this polar trophoblast, so mural trophoblast. So this is the blastocyst. Blastocyst is what will attach to the or implant on the uterus. So during the 6th or 7th day, this blastocyst will enter the uterine cavity, and it will be attached to the uterine wall, most preferably the posterior wall of the fundic region. Okay, so what is fundic region, that we should see, right? So this is the uterus, and this is the fallopian tube. Okay, I'm drawing only one side. It's the ovary. Now, this region is called fundus. So normally the implantation should happen on the posterior wall of the fundus, like somewhere around here. The blastocyst will come through this fallopian tube and goes here, and it happens to be implanted there. Now what happens, it doesn't implant on the posterior wall. A bit to this side, what happens? Nothing happens actually. Anything above this, or any implantation on the upper part of the uterus is okay. Any other part than the uterus, the developing embryo cannot survive. There are two types. Okay, one is extrauterine and one is intrauterine. So this extrauterine is also known as ectopic pregnancy. The ectopic pregnancy can be tubular, can be ovarian, can be peritoneal. So this peritoneal and ovarial are pretty rare, but the tubular is very common, and this tubular can cause the rupture of the tube and severe hemorrhage. Now talking about the intrauterine pregnancies which is happening other than the upper part of the uterus, it can be near to the internal os. If you see, this is the opening to the cervix, this is the internal o. Okay, so depending on where in relation to the os, the internal os, there can be class 1, class 2, class 3 type of abnormal pregnancy called placenta previa. Placenta previa. Okay, so let me ask you guys one thing. What we call the inner lining of the uterus, it is called the endometrium, right? We all know that, endometrium. But at the time of implantation or during pregnancy, the endometrium is called another name. It is called the deciduum. So you have heard about the deciduous forest. Yeah. What will they do? They'll shed their leaves during a particular season. Just like that, this layer of endometrial lining, or this layer of uterus, will be shed off if there is no implantation and there is no development of pregnancy. Now this deciduum has 3 zones. Okay, the zones are stratum compactum, stratum spongiosum, and finally stratum basal. So stratum compactum, stratum spongiosum, and stratum basal. Here what happens is, the blastocyst without the embryo, the exposed trophoblast will be without the zona pellucida, the exposed trophoblast will be there. The exposed trophoblast is very sticky and it will stick to this uterine wall. Why it is sticky? Because of the presence of some certain proteins like L-selectin. So this endometrial lining will have the receptors for this protein, the L-selectin, and they will combine, and it will initiate the process of implantation of the blastocyst into the endometrial wall or deciduum of the uterus. Okay, and by the end of day 6th to 7th, the implantation will be complete, and we'll move on to the second week of development. I also wanted to discuss about a clinical condition called the hydatidiform moles. What is happening here is, instead of a normal embryo, a mass of cyst forms inside the uterus. This mass of cyst can be benign or malignant. Okay, and it looks sort of a grape-like structure, and this grape-like fluid-filled capsules within the cyst is what gives the name hydatidiform mole. Okay, this hydatidiform mole is actually resulted from some chromosomal abnormalities. What is this chromosomal abnormality? If you consider the most common, complete hydatidiform mole, it is because of the zygote which is formed has just the paternal genetic information. Okay, how does that happen? When the egg had no genetic material and the sperm duplicates its own genetic material within the zygote after getting into the ovum. So how can this happen? When the released ovum or the egg does not have a maternal nucleus or the genetic material from the mother, The sperm which entered that egg cell will multiply its genetic material and forms a zygote in which there is only paternal genetic material. Now the problem is the paternal genetic material is what controls or contributes to the formation of placenta. Okay, here the trophoblast will be developed because it is what becoming the placenta, and the embryoblast will not be present, or if present will be very less. Okay, and this moles actually secretes a very high amount of human chorionic gonadotrophin, which can be used for the diagnosis of this particular condition. Also, we can use a D&C procedure, or the dilation and curettage procedure, to both diagnose and remove the cyst from that affected individual. Okay, so this is about the hydatidform mole. It's easy, right? So let us meet in the next video next week. Until then, bye. We are going to discuss about the second week of development. Okay, see you guys.